Hi, everyone, and welcome to my video, PCR variations. So in the first two videos of this week, I talked about DNA replication and then PCR, so polymerase chain reaction, um, how we're able to do DNA replication in a test tube. Well, it turns out that there are some variations of PCR that are basically the same process that I covered in the last video, but they have add-ons or special tweaks to them that are designed to address specific circumstances. So we're going to talk about those here in this video. So in this class, there are two specific variations of PCR that we talk about. And we specify these variations with extra letters that we add on to the abbreviation PCR. So there's RT-PCR, and qPCR. We're going to talk about each of these ones separately here. So let's start with RT-PCR. Now, for RT-PCR to make sense, we kind of have to dip a little bit into what we're going to learn in week 5. So in week 5, we're going to talk about what's called central dogma. So basically, so far, you've learned that DNA is the instructions for the cell, right? It's got genes in it that have instructions in them. Well, what are they instructions for? Turns out that there are molecules in the cell called proteins. And proteins do everything that needs doing in the cell. So you've already learned about a few, specifically enzymes, which are proteins that make chemical reactions happen faster. So enzymes like DNA polymerase or ligase, those are all proteins. So anything that needs doing in the cell is done by these big molecules called proteins. And it turns out that the genes in DNA are the instructions to tell the cell how to build the proteins. So each gene is the instructions for how to build a different kind of protein. And each protein has a specific job, like we've talked about. DNA polymerase builds DNA chains. Ligase sticks or fixes the nicks in the backbone by sticking two bases together. So they've got functions all throughout the cell for metabolism, for repair, for all kinds of things. So these things are very important. So the way that the cell makes proteins from DNA, there's an intermediate step because the DNA is locked inside the nucleus. And we don't want to take it outside of the nucleus where proteins are manufactured. So it has to make a photocopy of the DNA. Let me switch back to blue here. It has to make a photocopy of the DNA on kind of cheap paper, you could say. So DNA is kind of expensive for the cell to build. So it makes a photocopy of the gene that it needs to use in kind of a cheap version of DNA that we're going to learn about more in detail in week 5 called RNA. So you don't need to know what makes up RNA for this week. Just know that it's kind of like DNA. It's got bases, and it carries information, but it's just kind of like a cheap version of DNA that's disposable. So it makes a photocopy of the DNA to build RNA, and then it ships the RNA photocopy of the instructions out to the outside of the cell or to the cytoplasm of the cell, where outside the nucleus, where the proteins are constructed. So then the factories that build proteins that we'll learn about in week 5 read the RNA to build proteins. Now, the reason why I bring this up is because RNA is important for RT-PCR. It turns out that the PCR technique that we learned this week, that we talked about in the last video, only works on DNA. Taq polymerase, which is the enzyme that reads the original DNA strand to build the new RNA, or sorry, to build the new DNA strand in PCR, only knows how to read DNA and build DNA. So if we want to copy something else, we can't use Taq polymerase. And RNA has information in it. For one thing, most RNA, and again, we'll learn about this in week 5, but RNA, one thing that's different about it is after the cell photocopies the gene, it goes into the RNA and cuts out all of the introns. If you remember from week 1, introns were pieces of junk DNA that were embedded in the middle of the gene. So if this is my RNA and it's got like these introns, maybe I'll use a different color here. It's got these introns here in the middle, right? When we go to build the protein, we don't want these introns in the middle. You could think of them like ads in the middle of your recipe. You don't want to read or you don't want to incorporate whatever is being advertised in the middle of your recipe into the final product. We need to get rid of the ads, clip them out. So the cell will actually come in here and clip out these clip out the introns and merge the exons, the remaining pieces that aren't junk DNA, together to make a final RNA copy here. So that can be helpful for scientists to help us know where the introns are, because the RNA piece will be the gene, but without all of the introns, without all of the junk. So if I'm trying to make a copy of the actual coding part of the gene, then I don't want to PCR the gene out of the DNA because that'll give me the introns too. For example, if I were trying to move a gene into bacteria, which we'll learn about how you can do that in week 10, bacteria don't have introns in their DNA. So if I gave them a gene from a human, they wouldn't know what to do with it because they don't know how to remove introns. So I'd want to remove the introns first. And if I take the RNA version of the gene instead of the DNA version, then that conveniently doesn't have introns anymore. It's just got the important parts of the gene. But if I need to make a million copies of the RNA version of the gene, PCR is not going to work because Taq DNA polymerase only knows how to read DNA. It can't read RNA. And RNA kind of isn't very helpful for our purposes because remember, it's cheap, which also means it's flimsy. It doesn't last very long. Sometimes an RNA strand will only last a few minutes. So DNA is a much better storage mechanism, and we want to get copies of the gene in DNA form, but without the introns. So you need to be able to read an RNA copy and turn it into DNA. Another example of where this could be useful, of where this is important, is, for example, with COVID-19, some viruses don't use DNA as their genetic storage mechanism. Some of them, like the COVID virus, store their genetic information as RNA. So in those tests, if you guys remember from the pandemic, when they would take a swab and stick it up into your brain, right, to take a swab and then they'd ship it off to test to see if you've got COVID-19. What they're doing is PCR. They're trying to see if it's in there. So they put a pair of primers in there that are supposed to only bind to COVID's genome. And so if PCR happens, that means there was a COVID genome in there to amplify, which means you're sick with COVID. If it doesn't amplify, that means there was no COVID genome in there. The primers couldn't find anything to stick to. So PCR failed, and that means you're not sick. But the catch is COVID has an RNA genome. So first we have to convert the RNA into a DNA form so that we can PCR it like you're used to. And that's where RT-PCR comes in. RT stands for reverse transcriptase, which is a long word here. Reverse transcriptase. And reverse transcriptase is an enzyme that that comes from certain viruses like HIV. We borrowed it from them because it's very useful. Normally, the cell only has the ability to read DNA and turn it into RNA. It doesn't have the ability to go from RNA to DNA. It can't do that. Because remember, Taq polymerase, which is, or DNA polymerase 3 or Taq polymerase, depending on if you're doing DNA replication in the cell or in PCR, they only know how to read DNA and build DNA. They don't know how to read RNA to build DNA. So we can't go backwards and convert RNA into DNA. But reverse transcriptase is the key here because reverse transcriptase is basically DNA polymerase 3. That's essentially its job, except it has the ability to read RNA and convert it into DNA form. So instead of using, so with RT-PCR, we don't use Taq DNA polymerase. Instead, we use this reverse transcriptase in its place. So the PCR will function exactly the same as you're used to. You still have to put in primers. We're still building 5' to 3'. We still use heat to separate strands. We still go through the same 3 stages, the denaturation, annealing, and extension. The only difference here is on the first round, reverse transcriptase will come in here and it will read the RNA version and convert it into the DNA version. So then you're going, and then the remainder of the ones, reverse transcriptase is nifty because it can do both. It can either read RNA and convert it into DNA, or it can read DNA and build DNA. It's sort of like DNA polymerase 3 better version, 2.0 version, right? So it can either read RNA or DNA. So if I put just RNA in the tube, it'll read the RNA version, make a DNA version, and then it'll start reading the DNA version and build lots of copies of the DNA version. So from there on out, it's just plain old PCR. So the only difference here is we use reverse transcriptase instead of Taq DNA polymerase. And we start with RNA to convert the RNA into DNA. And then we do ordinary PCR to make lots and lots of copies of it. So that's how RT-PCR works, is all it's doing is it's reading the RNA to build DNA versions, and then we're making lots of copies of DNA. Okay, the other variation that we talk about this week is called qPCR. And qPCR stands for quantitative. PCR. Now, one little side point that you'll probably notice, when I wrote out RT-PCR, I used a capital RT and then dash PCR. But when I write qPCR, I just put a little q in front of the PCR. That's intentional. That's actually how they're written. RT stands for reverse transcriptase, but when you do the Q, it's not Q-PCR. It's little q PCR, no spaces, no dashes, nothing. It's just little q, capital PCR. I don't know why. Blame it on the English majors or something, I'm sure. Okay, so qPCR stands for quantitative PCR. What this one's doing is it's trying to give us additional information about quantity. Because if you remember, PCR, I do PCR and I see if I got a product at the end or not, right? We got millions and millions of copies of it. But all that tells me is I've got millions of copies of it and PCR worked. I don't actually know how many copies I have. So ordinary PCR is just a yes or a no. So for example, if I did ordinary RT-PCR, right? So I would if I did RT-PCR, I'm trying to diagnose you with COVID. I use the RT, the reverse transcriptase, to convert it into DNA, and then I make a lot of PCR products. And so I see that and I say, yes, you have COVID, right? Because PCR worked. But it doesn't tell me how bad your COVID is. It doesn't tell me, are you at the early stages of COVID? Do you only have a few of the virus, a few copies of the virus? Or are you like super, super sick? You're on like day 5 of COVID and you've got lots and lots of virus in you, right? It doesn't tell me that. It just tells me, do you have COVID or do you not? So if I want to try and give you a more specific diagnosis, I need more information about how many copies you have, because we can't tell that from an ordinary PCR. We can only tell, yes, you have COVID RNA, right? And there are other applications too, but there's an example that we can all relate to. So quantitative PCR can give us an idea of how many DNA strands were in there originally. How many DNA strands you start with. Because if you remember, in the last video, we taught you this equation, um, where to be able to calculate how many copies of a DNA strand you have after a certain number of rounds of PCR, right? Um, so it's the X times 2 to the power of n, where X was the number of starting copies and n was the number of rounds of PCR, right? Sorry, PCR. There we go. Well, this is great because I know the number of rounds of PCR I go through, right? I program that into the machine. I say, I want you to do this 30 times, right? So I know how many rounds it does, but how do I know how many starting copies I have, right? Because we don't, DNA molecules are invisible. How do I know how many copies I started with? Where do I figure out the X here? So quantitative PCR is intended to help us figure out the X. How many copies did we start with in the sample? And the way that works is through special chemicals that they add in to the mixture. So they add in special bases to there. So ordinarily, when you do PCR, you use DNTPs, which are the individual DNA bases building blocks so that polymerase has something to assemble a DNA strand out of. But in qPCR, we use specially modified DNTPs that have a little glowing light bulb on them. See how good I can draw a light bulb here. Okay. They have a little glowing light bulb attached to them. It's a molecule that they glued to the DNTP that lights up when you shoot a laser at it. Okay. So, but only when it's built into a DNA strand. So that's nifty. So when it's built into a DNA strand, this molecule lights up. So that's helpful because that means the brighter your tube gets, your PCR tube gets, the more copies of DNA you have, right? Because we're incorporating more light bulbs into the DNA strands. We're building, the more copies of DNA get, the more copies the DNA will glow. So the brighter it gets, the more copies of DNA you have in the tube. So that can be helpful because qPCR machines have an additional feature where they can shoot a laser into the tube while the PCR reaction is running and get live measurements of how many tubes there or how many copies of DNA there are in the tube second by second throughout the entire PCR process. And when you do that, it creates a graph that looks like this. So what's going on here in this graph, and again, you're not going to have to know how to read this graph. This is just to give you an idea of how this process works. So on this graph, we've got the number of cycles that have passed down here. So at this point, they've done 4 cycles of PCR and 8 cycles of PCR and 12, right? So the PCR reaction is running along the way and the machine is constantly reading it. Then up the side here is fluorescence. That's glow. So it's measuring how much the tube glows at each second along the way. So again, the more it glows, the more DNA we have in the tube. More glow is more DNA. So notice here on this particular example that I found on the internet, you would expect the way we teach PCR is you would expect on round 1, okay, it's going to double and then 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 it's going to double, right? And we just keep on going until all eternity, right? You can just keep doing rounds of PCR and you'll just keep getting more and more and more and more copies of DNA. Turns out it's a little more crude than that. The way it actually works, let's see here. Where's my eraser? There's my eraser. The way it actually works is you'll notice here a couple of things. One, the line starts late, and each of these different colored lines is a different PCR reaction that they did, and they just plotted them all on the same graph. So each of these is a different reaction, but you'll notice some of them don't even start making copies of DNA or discernible copies of DNA until like round 22. So the first several rounds of PCR, nothing happened. So we had the primers in there, we had polymerase in there, everything's great, but nothing happened. The reason for that is there's a lot less magic in the tube than we make it sound in class, right? If I've got, or the fewer numbers of starting strands of PCR I have in the tube, the less likely it is that a primer during the annealing step will actually find that DNA strand, right? It's a hide and seek one. If I'm trying to hunt a guy or if I'm trying to hunt one guy in a gigantic field, then it's going to take me forever to find him. But if I'm trying to hunt a person in a million people, I'm going to find a person really quick, right? So the more starting copies of DNA you have, the easier it is for primers to find a starting copy of DNA, right? So if we start out with only 1 or 2 copies, the primers might not find those starting copies for a few rounds. It may take them a while because during the annealing step, they simply don't have enough time to hunt it down. So it may take them a few tries to find the DNA strand and bind to it before the PCR can actually take off and start working. So we can use that because we can track what round it starts taking off on, and that can give us an indication of how many strands we started with. So notice the blue, or this blue line here, the PCR reaction started in round like 22, right? Whereas on the brown one, it started on round like 26 right here, right? So what does that tell us? Well, that tells us there was more starting copies of DNA in the blue one than there were in the brown one. This one has more than this one. sorry, and red would have even fewer because it starts even later, and gray would have even fewer. So if these were four individuals with COVID, I would say the guy in blue is more sick than the guy in brown because he had more starting copies. There are more viruses in him than there are in the guy that's got the brown line here or in the guy with the red line. So that can tell me information. So we can use that to diagnose because it can tell us quantity of starting DNA strands, not just do you have COVID or do you not? Now, notice one thing to clarify here uh, on the difference between RT-PCR and qPCR. RT-PCR starts with RNA. So let's pop back over here. RT-PCR starts with DNA, or sorry, with RNA. We go from RNA to DNA. qPCR, like ordinary qPCR, is DNA to DNA. It can't read RNA because we use Taq DNA polymerase in qPCR. We don't use a special polymerase there. This one, qPCR uses Taq DNA polymerase, not reverse transcriptase. So qPCR is exactly like ordinary PCR. The only thing we're changing is we're keeping track of how many copies of DNA are in the tube at any given moment. Whereas in ordinary PCR, we don't bother with that. We just use ordinary DNTPs. The reason why you wouldn't do qPCR in every circumstance is A, if I'm actually going to use that DNA down the line, it's not a good idea to have it glowing because that extra molecule that I add onto it makes it so that the cell won't be able to use that DNA if I were to give it to it and say build a protein from this. It's going to be like, you messed up my DNA. It's broken, right? So qPCR is kind of an end deal. We're not trying to make more copies to use them later. We're trying to just see how many copies I started with and get an idea there. The other reason is qPCR is way more expensive than ordinary PCR. So you only want to actually do it when you absolutely need to. Um, so sorry, I just lost my train of thought here. Oh, so yeah, so ordinary qPCR is just plain old PCR, but with glowing DNA, right? So even though in my example, I used the COVID diagnosis thing or as my example, keep in mind that RT-PCR is the version that goes RNA to DNA. qPCR is the one that goes DNA to DNA. So you may be wondering, if this is how we diagnose COVID, how do we use qPCR? Well, it turns out you can mix the two versions here. They've got another version that we don't really teach you in this class. This is just for curiosity's sake for you guys. There's another version that they creatively called RT-qPCR. And RT-qPCR merges the two. So you use glowing nucleotides and you use reverse transcriptase. So it converts the COVID RNA into DNA, and then you use the glowing nucleotides to build glowing DNA strands and track it over time. So if this version over here that I was showing as an example, if this was for COVID, they would actually be doing RT-qPCR instead of either qPCR or reverse transcriptase PCR. So they can be used together. But for this class, we're learning about them separately. So RT-PCR, you convert RNA to DNA first and then do ordinary PCR. qPCR, you are doing ordinary PCR, but keeping track of how many copies of DNA you have in the tube at any given moment. So that's the difference there. So thank you for watching this video. Feel free to reach out to me and ask me any questions. I am happy to help, and have a great day.